live. Is it visible? Yes. Go to the live stream now. It's live now.
Yes, it is.
It is. So shall we go ahead? So the first of all, a very good morning to all. So today we will just see the paper pattern. So for your information, the paper is for 2 hours and. वो बंद करो. You are here. So paper is for 2 hours, that you need to remember, and it is for 50 marks. So how this 50 marks is distributed? Is distributed in two type of practical. One is going to be the major part, and second is going to be your minor part. So in the major part, that is your question number 1, that you need to focus is you will be having a lot many option you can see, but the same will not be there into your practical examination. There will be grouping X, Y, like this. So this group is, whatever group you are in, so that will be written on your table. But the point to be remembered is you have to study all 4 or 5 options given in the your question paper. So the first option is dilute the given sample, estimate the OD using colorimeter. This is going to be your first practical for the 15 marks. So for that, what you will be doing? You will be taking, here it is clearly mentioned, you have to take the 3 dilution. The first colored solution will be there. What would be there? It is up to a examiner what colored solution they are going to give. So you have to take the colored solution in test tube number 1 ml. How much you are going to take?
1 minute.
After that you will be adding 9 ml water in that. All right, then after that you will be filling it, and after that 1 ml from test tube number 1. You all remember the diagram which I have drawn in the regular practical from test tube number 1. Can anyone show me that diagram, please? Fast. Thank you. So this is now see, in regular practical we have done major dilutions, but here we will not going for it. So this is distilled water 10 ml, so that is your blank. Okay, in test tube number 1, what you have done, you have taken the 1 ml colored solution, right? Now 9 ml water was there. Now from this solution you are going to take 1 ml in test tube, next test tube, that is going to be your D2, that is your second dilution, and after that you will be adding 9 ml water in that. Got my point? Now see, here you have shown with arrow 1 ml solution. So here better if you will write 1 ml the colored solution is there from the test tube number D1, and 9 ml distilled water. Is that clear? Understood? Now in the next you will be taking from D2 1 ml colored solution and 9 ml distilled water. Now see, your test tubes are ready, 3 dilutions are ready, D1, D2, D3. In blank you have taken only and only the distilled water. After that, what you will be doing? After that you will be taking the OD. Now how to take the OD from the colorimeter, that I had already explained into the regular practical, but I will add the link how to take the OD from the colorimeter. That video is already been there on the channel, not going to show it again. You can take the from that particular video. I will add the link. You have to remind me to add the link in that. Okay, so let's move on the next practical. Calculate concentration from given OD by the formula. Now here 3 concentrations will be printed and given to you with your question paper if this particular question is asked to you. Is that clear? So now concentrations will be given to you. You have to find the standard, calculate the concentration. So what is the formula for the calculating concentration? OD of unknown divided by OD of standard into concentration of standard. With that formula you have to find out 3 concentration. That is a very easy question, we will not discuss on that. Now find pH of water sample and comment on their chemical nature. This is important question. Now see here, here you have to find out the pH. So how you all are going to find out? We have learned lot many methods. What all methods we have learned? Universal pH indicator, cabbage indicator, as well as we have learned from the your litmus paper. You can specially say it as a pH paper. So if it is pH paper, only you need to dip it, right? Only dip, and whatever color you are obtaining, you have to match it with your standard color solution. Please come at this side. I will not be able to see this side. Okay, understood. And you also, you all can come here. And next thing, when it is universal indicator, you have to add the universal indicator directly into the solution. Okay, that is simple. Red cabbage indicator में 5 ml. Next question, red cabbage indicator 5 ml of any type of the solution. You will be adding the 2 or 3 ml whatever is given into the protocol, that you will be adding, and you will be observing the color change. Now here, point to be remember, in the case of the red cabbage indicator, you all need to remember the color. Okay, you have to by heart the filters. Is that clear? Chalo, let's move on to question number 2. Question number 2, perform the experiment for the separation of pigment by adsorption chromatography by using chalk. Right, we all have done. You have to make the baseline, and after that you will be drilling a simple depression into the chalk, and after that you will be adding the two dyes or sketches, whatever is mentioned that particular time you will be adding, and after that you will be keeping it into the beaker, and you will allow the solvent to run, and after that you will be just simply getting the result. And don't forget to draw the chalk as we have drawn it into the regular practical. Please open that. That is important in exam, okay, because in observation you have to draw it. That's compulsory. Chalk chromatography, it is important to draw the chromatogram. Yes. So this way you will be drawing the chalk, which we have done into the regular practical. You have to take the size of the chalk, then after that you will be drawing where you have drawn the baseline, then depression also you have to mention, and where you got the pigment separation. Is that clear? The most important thing, don't forget to mark the distance traveled by the solvent into the wet chalk, because once the chalk is dried you will lose that point. Is that clear? So it is very important, when your chalk is wet, that time you will draw. And the another precaution you must take care is, the when the chalk is wet you will handle it very carefully, it should not break. Is that clear? Chalo, that's it. Let's move ahead. Open next, that amino acid paper chromatography. Perform experiment for the separation of mixture for amino acid by paper chromatography. In that also you will be drawing the chromatogram. Now see here, amino acid will be given, a chromatogram will be given of Workman filter paper, and that chromatogram you have to draw like this. Okay, you will be drawing it, and after that where you have drawn the readings of that reading. Yeah, hello ma'am, see this one. Here you have not drawn, but you have to draw the reading wherever you are getting the separation line after. Now see, the most important thing, when you will be using the neem hydrin, you need to remember the neem hydrin is a neurotoxin. You have to handle it very carefully. Is that clear? So while handling neurotoxin you all need to be very careful. Then next is perform thin layer chromatography. So see, this is same as paper chromatography for performing. Only the thing is that what the thing you are doing on the paper chromatogram, here you will be doing on the TLC chromatogram. Is here you will be just performing it. You will not make the TLC plates here. Okay, you will be getting the ready-made TLC plates for your examination. So we'll not discuss on that now. Next, question number 3. It is a 5 marks question. You have to focus the slide under the 10x 45x and show it to the examiner. So here permanent slides will be given to you. You will just observe it under the microscope, and whatever diagram you are observing you have to draw it on your paper, and after focusing immediately call to the examiner and show it into the 10x and the 45x. Is that clear? Let's go ahead. Calculation part is already done, and see, this calculation video is also okay, so you can see it. You will get it into the semester 2 playlist. So all the central tendency problems are there. This is what I remember. If it is not there, I have already explained it into the regular practical. You have a doubt, I am just a phone call away for my student, not for all. Okay, now identification, point to be remembered. In the case of the identification, no one have to draw any diagram. Is that clear? Focus the diagram given. Now in identification you will be getting two safety symbols. Now safety symbols, you have to read the property. In theory also we have studied, in regular also we have studied. So only two safety symbols, then part of compound microscope. So for this, a microscope will be given, and that particular part will be labeled identify me karke. Is that clear?
Yes.
And the next is transgenic animal will be from your textbook only, but DNA fingerprinting problem will not be the same given into your textbook. Only the thing is you should understand basic concept of identification of DNA fingerprinting. You all know that you have to just match the bands. If whatever band is matched, you have to write answer in your word. So 1 and 2 example we have done into the regular practical, same type of problem will be there. Understood? Problem will be of same type. You will be given the electrophoresis, whatever observation table will be there, and after that you have to identify it. Is that clear?
Yes.
Then your neat, clean, properly certified journal have 5 marks, and it should be covered also, black cover as suggested. And after that there will be a viva voce, but point to be remember, the viva voce will be based on the practical component, not the theory. So this is all about your paper 2. If anyone have any doubt, will ask me.
Okay.
Yes, any doubt?
No.
So shall I stop now?
Yes.
Just a minute.